From Microbe TV, this is Immune, episode number 76, recorded on January 23rd, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, everyone. It's great to be back. It's January.
Almost done.
Yeah.
It's crazy.
Well, you know what? The year will be over, Cindy.
Oh, no. Don't rush it. Too many fun things to do. Too many good immunology papers to review.
Also joining us from Cleveland, Ohio, Steph Langle.
Hey, everyone. Glad to be back. Had to miss the last one, but very excited to be here today.
Did we have a guest on the last one?
We did, yeah.
Oh, that was really cool.
Yeah.
That
Yes.
was a cool one.
I listened to it. It was very good. Yeah.
Cytokine Dictionary. Yeah.
Yep.
From Madison, New Jersey, Brianne Barker.
Hi. Great to see everyone. And I'm excited about a new year of new immune stuff. Yeah.
Stuff. It's a highly technical term, stuff.
You know I use a lot of technical terms like that.
Yeah, it's good. Keep it accessible.
Yes.
We have a guest today all the way on the other side of the coast. Most of us are near the East Coast. Steph, you're pretty close, right,
Pretty close,
to the
yeah.
East Coast. And, of course, Cindy and Brianne and myself are right on the East Coast. But our guest is from California, Riverside, California, the University of California. Welcome, Juliet Morrison.
Thank you so much. Hello, everyone. I'm really excited to be here on the show. So, hi, listeners.
I'm very excited to have you for reasons that will become evident in a few moments.
Indeed.
Before we start, Cindy, did you want to mention Solving for Science again, or was that?
I didn't have it open right now, but yeah, I can just off the cuff. Any immunologists interested in learning more about Solving for Science or about the Discovery Stack, which is a really interesting, it's like trying to tackle huge problems in publishing. And the way they're trying to address this is maybe individual figures or individual papers being published by a different mechanism and not always by the journals that want to extort a lot of money from us. You can find more at solvingforscience.org. So I just want to double check.
Solving4.org.
It's solving4, FOR.org. And that you can log in and find out more information. But we're recruiting immunologists to participate in a pilot program where you're going to either submit your paper that you can submit to a regular journal, and you're going to see how the review process goes through this new mechanism as well as the original mechanism, and they're going to compare that. Or you could even volunteer to be one of the reviewers that's participating in this program. So go check it out.
If you like what we do on these science programs, Immune and TWIV and all the others, we'd love to have your support. We're a nonprofit organization, so you're in the U.S., it would be federal tax deductible. Go to microbe.tv/contribute. Juliet, let's start with your history. As far back as you can remember, I like to say.
As far back as I can remember, well, I guess I would say that would be, kindergarten, but maybe that's a bit too much.
Were you interested in science then?
No. I was not interested in science then. So I'm from the island of Jamaica. I was born in Kingston and raised in Kingston as well. And I got interested in science in, I would say, high school. I got this opportunity to attend this scientific fair in where was it? In Britain, and managed to actually see that people could do science for a living. Before that, I didn't even really think about it. I just didn't. You just don't have those role models, so you don't know. Yeah. And realizing that got me very excited about biology and chemistry. And so I decided to work on that during high school. And at the end of high school, I wanted to go see something new. And explore a different countries. So I actually started looking for colleges abroad, and mostly looking in the UK, but I actually typed in scholarship, science college, and Bard College popped up, and they had this science scholarship called a Distinguished Scientist Scholars Scholarship or something. And it was my gateway to America. And I got to Bard. I started doing research in my freshman year, and I loved it. And I got a great education there. So I felt very prepared to go to grad school, and I was encouraged to apply to Columbia and to some other places. I ended up deciding on Columbia because I was really, really interested in this one lab that was doing some stuff with interferons and poliovirus. And that happened to be Vincent's lab. So Vincent is my PhD advisor, and I was in his lab for 6 years, and we did some really cool stuff. Yeah.
Yeah,
So
we
what-
had a good time.
What type of work did you do as an undergrad? And then what type of work did you do with Vincent?
Oh, so as an undergrad, I did some summer programs where I was in a virology lab, but I wasn't doing virology. I was just I was doing sequencing of some stuff. So I didn't learn how to do anything with viruses until I actually got to Vincent's lab. And I had a summer project at Rockefeller where I was looking at was B cells and somatic hypermutation. And that project worked out kind of well. I think I don't remember it quite at this point, but you know, I learned a lot, and it helped me get into grad school. And so when I went to Vincent's lab, I really wanted to work on, initially I wanted to work on dengue virus, because dengue virus is a big deal in Jamaica. But I also fell in love with poliovirus because it grows so well. It is like the best virus. It plaques really nicely, and it's just a good model virus. So I worked on poliovirus, and I showed that the 2A protease of poliovirus confers this ability to replicate in cells that have already been treated with interferon. So it might be antagonizing an ISG or something like that. We never did quite get to the mechanism. I had to graduate, but it was good. It was a good time.
The big
Wow.
question is how many plaque assays did you contribute to the wall of
Yes.
None of those are actually my back assays. He started the wall, I think after I graduated. Or
Oh.
maybe, yes. Yeah.
Yeah, that was just one experiment by a postdoc years later.
Wow.
Oh, really?
Yeah.
Oh, I didn't realize that was one experiment.
So the experiment was we were collaborating with Charlie Rice and John Scoggins at Rockefeller, and they were taking every virus and doing ISG screens to see which inhibited.
I remember that paper.
Me too.
And we were one of the few labs that actually did an infectivity assay rather than a reporter gene
Sure.
assay. And so that was a big experiment to confirm one of the ISGs, particularly interesting, which I can't remember anymore, but she did many concentrations and many dilutions and many time points, and it ended up being 1,600 6-well plates.
Wow.
And so when she was done, she said, "What do I do with these?" And we built a wall out of it.
oh, wow.
That's
But I have to
awesome.
say, when I left my office. Well, I was kicked out of my office last year, and they tore it down and threw it out.
Aw.
No way!
It's
That's
gone.
so
It's gone.
sad.
Yeah, way.
Yeah,
Yeah,
it's okay.
bummer.
You know, sometimes art pieces have a limited. I viewed it as an art piece actually, and it had a limit. Someone said you should donate it to a museum. I said, I'm not dealing with that. Anyway, yeah, but the wall wasn't there when you were there, Juliet, right?
No, but I
Yeah.
came back to visit once and took a picture by the wall. So
That's right. That's right. Yeah.
Now,
Cool.
Juliet, during your time there, when you're going through your PhD, what were you feeling in regards to your career trajectory? Did you like academia, and that was the path you saw for yourself, or were you still trying to figure it out?
I was still trying to figure it out. I went back and forth. Academia is so hard, and you pick that up very quickly, that funding is going to be an issue, that the hours are long, and that you're always writing grants. And so that kind of worried me. So I explored other opportunities too. I took classes at Columbia Business School. I went on a lot of informational interviews, and I just still couldn't think of anything that I'd rather do than academic science, unfortunately. And so by the time I was finished with my PhD, I was pretty certain that I wanted to go into it.
And you're right that those definitely can be some of the impediments to academic science. But I would say on the flip side, and would like your opinion too, I think the benefits of it, of course, are, you're discovering new things. You own the ideas. I mean, in the sense that these are your ideas that you can come to fruition.
No, absolutely.
And I find that, the work-life balance thing, I love that I can decide to leave at 2 or 3 to go pick up my kid and then finish the grant at a different time. I love that flexibility. So I don't know if you feel similarly, but I always like to kind of bring up the positive just so trainees know.
Yes, trainees should know. You're absolutely right, Steph. It is actually a pretty flexible job. Yes, it is a hard job, but it also gives you that level of flexibility, and it's intellectually challenging, and it's really fun to have ideas and to test those ideas and to actually see them come to, fruition. Like, I love that stuff. I love it.
And mentoring students and training.
And mentoring students is awesome. I just graduated my first PhD student, and I'm so psyched.
Oh, yeah. Awesome. Congratulations.
Awesome. Thank you.
So I know it was a letdown, but what did you do after my lab?
I then went to Adolfo Garcia Sastre's lab over at Mount Sinai. Nice. And even though at that point he was mainly focused on influenza virus, I decided I wanted to work on dengue virus because he also did some dengue work. And I ended up isolating this protein called UBR4, which is this huge 600 kilodalton protein. I found that it was important for dengue virus to antagonize interferon signaling. Specifically, it seems to help the NS5 protein interact with STAT2 and degrade it. Yeah. Well, not NS5 degrading it, but it gets degraded.
Okay.
So
Yes.
I've noticed that, the paper that we're going to talk about today talks about influenza, which, of course, you might have heard about in Adolfo's lab. And in fact, I now feel very curious about what happened to your love of dengue.
It's still there.
And what's going on with you and dengue?
I know. And it's like my baby. So after Adolfo's lab, in Adolfo's lab, I worked on dengue and I worked on yellow fever virus. We found this really cool mechanism that it uses to antagonize interferon signaling. But I also, at that point, people were starting to really talk about transcriptomics and big data and all this stuff. And I really wanted to get training in that area. So I ended up going and doing a second postdoc in a lab, in the Cates lab at University of Washington in the microbiology department there. And the project I was put on was an influenza project. And so I started getting into influenza. And it wasn't very hard to pick up on the influenza stuff because I had spent so many lab meetings listening to influenza project presentations and stuff when I was in Adolfo's lab. So it somehow passively just entered my brain, and all of a sudden I'm like, wait a minute, I know a lot about influenza virus, at least more than some people. So yeah, so I got into it, and then I got into transcriptomics in general, where I was just like, I think, I love thinking about how our bodies respond to pathogens and how that can be modified in some way as a therapeutic strategy. So I wanted to continue, since I did an influenza virus project in the Cates lab, I wanted to continue with that theme because I got really excited about these signatures that we picked up, and I really wanted to figure out what cell type it was coming from. And so I still love dengue, and I have a dengue project, but I don't have a student to work on the dengue project. So that's but there are some ideas that I have that I'd really like to follow up on.
Good. I'm glad that you didn't let your love for dengue go away.
Thanks, Brianne. No,
When
it's
did
still
you,
there.
When did you go to Riverside? What year?
2018 is when I October 2018 was when I started there, and
Became a West Coaster.
I did. So far so good. It's I mean the heat is unbearable in the summers, but winter is really nice. So I like that, and I found my colleagues to be pretty collegial, and I have some collaborations going. I teach 2.5 classes a year, so it's a bit heavy on the teaching load. To also do research, but I found it rewarding teaching undergraduates
What do you
and
teach?
mentoring grad students. I teach host responses to viral pathogens, which is this course that I developed. It's basically a viral immunology class and for upper level undergrads. And we basically learn about immunology by looking at each viral disease of interest and just, working backwards. And they love it.
That sounds amazing.
I was going to say, I want to take your course.
Yeah, can I take that class too?
Sure.
Can we register?
Yeah, it's really fun, and I give them this challenge of coming up with host targeted therapies for things like Ebola and influenza, and some of the answers are really sophisticated. Like, I'm really impressed. So that has been really surprising to me how much I actually enjoy undergraduate education. Like, it's really fun to see them get it and get them excited about science.
What's the other course you teach?
Comparative virology. So introduction to comparative virology, and it's very standard. Let's study the replication cycles and the structures and compare them across the different viral groups. And I'm teaching that currently, right? Yeah.
Great.
Yeah. Mm-hmm.
All right. Well, a couple of months ago, you emailed me. You said, "Hey, I published this paper. Maybe you'd like to do it on one of your podcasts." And I said, "Well, why don't you come on?" And so here we are. It is a PNAS paper. Pleural macrophages translocate to the lung during infection to promote improved influenza outcomes. So, Juliet, tell us about the authors on this paper and what they did.
Okay, let me pull it up here. A lot of people on this paper. Okay, so the first author is James Stumpf. He is he's a graduate student who just defended recently, and he is the one who drove this project and did all of the immunology. Sang-Young Kim was a PhD student. He's now defended as well in the lab of Mira Nair, who is one of my collaborators here at UCR. And let's see, Matthew McFadden. Okay, wait, let's go to Adriana Ferrero. So Adriana Ferrero and Andrew Nishida were the people who did a lot of the transcriptomic analysis initially. And that was when I was back at University of Washington. So this project has been coming from a really long time. I've been really mining this data set. So now Adriana Ferrero now has a lab. I think it's in Ohio.
She's at Ohio State.
Yes, Ohio State. Mm-hmm. And then I have Roxana Shirazi, who is a grad student in my lab who helped with mouse infections, etc. Irit Gatviks is a professor in Tel Aviv at Tel Aviv University, and she's the one who came up with this tissue deconvolution algorithm called DCQ, which we used, and she and her student Yael Stierman were helpful they were helpful at explaining the limitations of DCQ and what we could do and not do with it as a predictive tool. And so that was a good collaboration there. And I think that was everyone.
Yep.
No, Matthew McFadden is in Adriana Ferrero's lab. Yes. Okay.
So I like this paper because there's transcriptomics, but then you do some experiments, wet
Yes.
experiments, right? We often see so many transcriptomics papers, which are just data dumps, and you don't know what it means. So I like that you test your hypotheses. So tell us the background. Why did you do this?
So initially, some years ago, I was interested in figuring out if there were any signatures that could separate a mild influenza outcome from a severe influenza outcome. So so this was a collaboration actually with Terry Tumpe's group at CDC some time back, where they had infected mice with different strains of influenza, and we looked at the outcomes, mostly weight loss and mortality. And we also harvested the lungs, and we did transcriptomics on the whole lungs to identify differences between the mild outcomes versus the severe outcomes. And by mild, in this context, I mean that the animals survive the infection, and severe, they die. So very, very, binary. And then there was another paper that was similarly designed to that paper, where it was also looking at different strains of viruses and looking at the whole lung transcriptome. And so we put that together, and that served as the basis for our predictions for this paper. And we were able to see that we were getting this when the animals recovered from flu, when they recovered from flu, they would have these increased expression of these coagulation genes and in some other pathways, but this was completely absent in the animals that succumbed to infection. And we saw some other signatures there, and because the lung is so complicated and there's so many different cell types, we wanted to figure out which cell type this could be. And that's when we started thinking about using a tissue deconvolution algorithm like digital cell quantifier to look at our data to see if there are any populations that differed between the animals that died or versus the animals that lived. And by using that tool, we were able to pick up some patterns that were already known in the literature. Like it predicted that there would be more inflammatory monocytes in the animals that developed severe disease versus the animals that had mild disease, which is something that people have seen multiple times. The neutrophil prediction was also a good one. So we thought, this is a great tool. Let's see if we can actually use it as a discovery tool. And we were able to, and by profiling all of our data, we found that in the animals that had the mild outcomes and survived, they were predicted to have this cell type that resembled small peritoneal macrophages. And we found that to be pretty curious. Mm-hmm. And so that's where the hypothesis generation started. So we said, okay, peritoneal macrophages, what are these anyway? So the peritoneal cavity is a serosal cavity, and there are 3 serosal cavities in our body. There's the pleural cavity, which surrounds our lungs, the peritoneal cavity that surrounds our abdominal organs, and the pericardial cavity that surrounds the heart. And because the lungs, we were profiling lungs, we figured that maybe this cell type is not coming from the peritoneal cavity, but instead from the pleural cavity, since they actually have very similar immune cell populations. So we have macrophages, small macrophages, large macrophages, some B cells, some T cells, and these are found in all of these 3 cavities.
So, Juliet, can I clarify? So this, serous membranes I'd never heard of before. Can you imagine serous, SEROUS? But I've heard of the pleural cavity and the peritoneum and all that. So in the lungs, the pleural cavity, is it actually kind of like a pillow between the lung and the rib cage?
Exactly. I consider it to be a fluid-filled cushion, basically.
So it's fluid-filled, yeah. Mm-hmm. How big, how thick would it be roughly? An inch or so or less?
In humans, I have no idea. In mice, it is super duper. It's super duper thin. So It is about a millimeter that you're of space that you're working with when we're trying to inject these mice.
And so it's full of fluid and also obviously has immune cells in it, right?
Yes. Mm-hmm.
And is this like a repository of immune cells from which they can go and take care of infections? Is that the idea?
Yeah, that's what has been shown these days, is that they can serve as a source of macrophages for the lung.
Right. And so we're talking about the area outside of the lungs, but inside of some other connective tissue.
Yeah. It's a special fluid-filled space that is important for breathing. It keeps your lungs intact when you breathe, and that used to be that's what was known best about it. That it offers this protection, but I think it's really cool that it also offers this additional layer of protection too immunologically.
So I did a quick search, and it says that the normal thickness is 0.2 to 0.4 millimeters. So it's super thin, and it's about 5 ml of fluid in the pleural, whatever, in a healthy human. So
5 ml of fluid?
5 milliliters of fluid.
Oh, okay.
Okay.
I was like, I don't know what that is.
I said milliliters of fluid. Maybe I spoke really fast. Sorry. It's not much.
No, it's not a lot at all.
No.
Do the macrophages come out of the bone marrow and then differentiate there, or are these a different type of macrophage?
Okay, so there are two types of macrophages there: the small ones and the large ones. The small ones are from the bone marrow, so they don't last as long, and they're replaced by monocytes as time goes by. And some papers have shown that the small macrophages can turn into large macrophages, but that might be a little less. I think there's some controversy there, not controversy, but there are some disagreements there as to where exactly the large pleural macrophages are coming from. But we do know that they are seeded, that they are a long-term population, and it's only when there are issues that they are replaced.
And so how does that fit in with the M1, M2 phenotype of macrophages?
So from what our limit, we've done some nano string with a collaborator, and what it looks like is that they have more of an M2 look, both of these cell types.
Okay. And for our audience, macrophages are very plastic in the way that they can differentiate into different types. And it's not always clear that even this M1, which is thought of as pro-inflammatory, and M2 is anti-inflammatory, but that in itself is probably way oversimplifying it. But we have to try to categorize these things so that we can define different responses. And so that is what I was referencing here in regards to the pleural macrophages. And right, so they may, Juliet, you're saying be more M2. And without infection, we think they are maintaining homeostasis. We think they are helping to clear debris. Like
yes.
this.
Yeah,
okay.
Exactly. So I think that they're in there just maintaining things, picking up on any infections or cancer. Mesothelioma is a big one that affects the pleural cavity and the peritoneal cavity too, I think.
Yeah.
Yes.
Okay.
So Julia, could you tell us how you used the transcriptomics to identify these cell types? How does that work?
So basically, DCQ is based on, it basically puts together two pieces of information. So it takes the transcriptomic data from ImGen. So ImGen is this big source of this big consortium, and they have a whole bunch of microarray and now RNA-seq profiles of purified cells. So they have hundreds of these profiles there. So you can see B cells that came from a mouse that got a viral disease, or you can get T cells or macrophages from a mouse that was stimulated with LPS. And so you just have all of these cells and all of these profiles. And what DCQ does is to take your gene expression data from the whole lung or any whole organ, and it does some sort of magic. I did not develop this, and manages to predict what cells might be in your organ, based on the comparison between the two data sets.
So you had lung RNA-seq from severe and
this is
and
actually lung microarray data. So this is way it's like some of the data are actually quite a bit old, but they were clean and very well done experiments. So I like when I know that the data I'm using came from something that was really rigorously done.
Right. And so from these data, then you're able to pinpoint these signatures that are happening in response to infection.
Yes.
And I don't know if you want to talk. I mean, it's really interesting to look up. First of all, I love combining these two different studies to try to get more data out of it. Because, we publish, studies from your lab or one lab and with a set of animals, but you want to be able to, over time, combine all of that so we can just get more from, taxpayer dollars and also all of our labor. And I love that you did that here. And you've really show, like I'm looking at figure 1, you're describing to us these different mechanisms and pathways that are being upregulated depending on the disease severity, and you infected with a variety of influenza strains. And so can you talk a little bit about how you thought about that, combining these studies together with the different influenza strains and what you anticipated?
So the first study that we used here and used for DCQ was from a study that I was the first author on, but it only had 4 strains of flu. 3 of them were avian derived, and 1 was the 2009 pandemic H1N1. And I was seeing differences between the avian and the human, but I only had 1 human strain. And so even though the avian flus were causing severe disease and the human strain was not, I think it was just a bit underpowered, because I wanted to also see if I could throw in some other H1N1s in there that were that caused different levels of disease. And so that's where we came up with the idea of combining these two studies and trying to see a bigger picture.
Right. The different levels of severity.
Exactly.
Which is
right. Yeah. So we have some really intermediate phenotypes too. So I found that was helpful in really looking at those signatures.
Right.
And so overall, was the response mostly similar across all of those, whether they caused severe or mild disease, and then there were just some unique signatures, or were they really very, very different?
They weren't that different. So for the ones that were very severe, the severe outcomes, they looked very similar to each other, and the ones that had the very mild outcomes looked very similar. And then the ones that were intermediate, you see this sort of intermediate level of the various, upregulation of genes or downregulation of genes. So that's more intermediate phenotype.
And were there specific pathways that stood out to you?
Yeah, so I was very interested in the we saw lipid metabolism and coagulation signaling.
Yeah.
We saw that genes in that path in those pathways were upregulated in the animals that survived. And lipid metabolism is very hot right now. But for me, I was excited about it because that I was reading a lot of papers about macrophages at the time, and that's a big thing in macrophage biology. So, so we decided to follow up on that. And there were some other pathways that looked interesting too, metabolic responses, tissue repair mechanisms. It all felt very macrophagey to me, if that's not a word, but That's what we thought we were going to get, but I never would have imagined that it was going to be a pleural macrophage. So,
at what kind of timing did the severe versus less severe infections sort of distinguish themselves in terms of transcriptomics?
So from the transcriptomic studies, we found that we were getting these differences very by day 4. Day 4, you'd see a lot of weight loss, and then for some of the animals, they'd have to be euthanized because they meet that criteria.
Right.
So you have animals with mild disease. You have they're associated with these peritoneal-like macrophages, right? And then serious disease, less of those cells. Was
right? Yeah, that was a prediction. Less of those cells or none of those cells.
Interesting.
Okay. And then you actually did wet experiments to test it. So and you called them wet experiments, which is great because sometimes I call them experiments, and then the transcriptomics people say, "Well, we do experiments too." Okay, so tell us about the wet experiments.
Oh, okay. So, so we came up with that hypothesis. We're like, okay, maybe it's pleural macrophages that are getting into the lung. And I was very interested in testing this, but I'm not actually, a trained immunologist. So it took quite a lot of effort to get up to speed on what all of those markers are. And I'm still trying to figure out all of the markers, but I ended up picking up some flow cytometry techniques, and I wanted to keep it very simple because I don't consider myself an immunologist, really. So I wanted to. I figured that maybe the small pleural macrophages were getting into the lung during the stage of recovery, but I didn't know how to distinguish that because there's so many markers for macrophages that overlap between different populations. Right. And so I decided, why don't I try to tag the macrophages, the pleural macrophages, in the pleural cavity with a special phagocytic dye and do that experiment and see if the macrophages then end up in the lung. And that took a lot of development because targeting that space is really, really hard. But we were able to do it. And we got pretty consistent data showing that a small population of these labeled cells would end up in the lung. Yeah.
So you use a dye that's taken up by
yeah. So it's
any phagocytic cell.
Any phagocytic cell. Yeah.
And did you inject it into the pleural space?
Yeah. So we inject it into the pleural space with this tiny little modified needle. Then we rest the animals for a day, and then we infect them with influenza virus, and then track them. And so
I'm very impressed with whoever did this injection.
Oh my God, let me tell you, my former student James is just a king of it. Like, he is better at it than I am. And I'm so sad that he's gone because right now, it's just not as efficient.
Well, Juliet, that's the goal, to make people who are better than you are. That's what I did with you, right?
Right? Oh, come on, Vince, please. But no, I'm really proud. Like he when he joined my lab, he had never actually done an experiment before. Wow. So he went to grad school without doing summer research or anything because he always worked throughout college. But he was very smart and he picked up very quickly. And another thing that's really important for this story is that James Stumpf is Native American, and that is a population that rarely ever get PhDs. So it's 0.5% of doctoral degrees conferred are to indigenous folk of this country. So I'm super excited about this. He now has a great job at 23andMe, and it feels good. Yeah.
Oh, that's great.
I think you make a great point in mentioning that, he had to work through school, so he didn't have an opportunity because it's admission season and people are evaluating applications, and it's such an easy thing to just say, oh, that student has a paper, they're higher than this student,
Right.
without holistically looking at the application and figuring out like what opportunities did they have, because you might pass over a student like this who's phenomenal.
Yeah.
And that's just such a great story. I love that.
Thanks. Yeah, I'm super proud of him. And yeah, we really should consider people more holistically. It's really hard, of course, but I think, we're missing out on a lot of talent for the people who didn't do research in undergrad.
Yeah, it's more work, but it's worth it.
Yeah. Anybody could be good if given the opportunity, right? You just don't know who it, and just because they have money or something doesn't mean that they're the only ones, right? So we really need to think more broadly.
Yeah.
And just as a side note, I think it's going to get harder because I'm hearing across the board 20 to 30 to 40% increase in applications to graduate school this year. I think, at least, I don't know about your institutions if you're seeing that.
To our PhD program, yep.
Wow.
Right. So I think when that happens, there's only so many people who can review these things. So it gets harder to then down select. And so you have to use metrics, but you're right. We have to keep in mind, how can we holistically continue to drive that home? Yeah. I'm glad to hear about your student though. That sounds like an interesting job to work at 23andMe.
Yeah.
Yeah.
So Julia, when you put the dye in the pleural space, did you look at other macrophages to see outside circulating?
Oh yeah, we did. So we looked at macrophages in the lung, and we also looked at monocytes in the blood, and we looked at the lung draining lymph nodes as well. And we saw no dye in those compartments.
Okay.
Interesting. And we'll talk about the depletion experiments, but and maybe this question can be answered after you get work through your experiments. But I'm just curious the interaction happening. So influenza, we know, binds to sialic acids and is replicating in the lung, but what is its relationship with the pleural space? Do you see high titers there? Is it?
No, we are not getting any viral infection in the pleural space.
Got it. Okay.
Yeah, but we do see an increase in inflammatory cytokines in that fluid. So there is communication there.
So then if the virus isn't there, but the macrophages, so then we're not really thinking that the virus is directly interacting with virus-infected cells. It's more trying to maybe downregulate the inflammatory response.
That is what I think is happening. Right. Yeah, right. I think they come in and they offer this anti-inflammatory role. Yeah. Which
is interesting because we talk a lot about you have to have your immune response be effective and quick, but if you cannot downregulate it, you are going to struggle. You're not going to. You're going to have higher pathology.
So then what ends up being part of the difference between the severe and the less severe infection?
In what sense?
Well, so is it a difference in the inflammatory cytokine in the pleural cavity? Is it a difference in how the macrophage responds to the inflammatory cytokine?
I think it's all cytokine mediated. That is what I think is happening here, and we just need to figure out which cytokine it is. So I've designed some experiments, but I'm waiting for funding to do these experiments because I'd really like to figure out what exactly is happening there. And we're doing some experiments now where we are just doing setting up an inflammatory environment in the lung without it being an infection. So using LPS or poly-IC, and still trying to figure out the data from that right now.
So Juliet, the influx of these labeled macrophages is different when you use two different strains, a lethal and a not, right?
Yes.
So is the cytokine profile different with those two?
Yep, the cytokine profile is different. So you have a lot more inflammation with so we use two strains here. One was the Netherlands 2009. So this was isolated from somebody in the Netherlands, and we have one isolated from somebody in California, and they're actually very, very similar, but they differ by about 5 amino acids, and they behave differently in both Black 6 and Balb C mice. So the California 09 strain was our mild strain, and the Netherlands actually ends up killing the animals at a certain point. And so we use those as a comparison for each other, and you can see that the inflammatory cytokines are so much higher with the Netherlands 09 strain. in both the lung and in the pleural cavity.
All right, and you think this is mediated by cytokine differences, right?
I do, yeah.
And so someday in the future, you're going to inject mice with specific cytokines like our last guest.
Right, yeah. I mean, I'm hoping that we could identify something and then actually do that proof of concept experiment. That would be really neat.
I mean, it's probably going to be a mixture, not just one.
Not just one, but, if we could find something pretty dominant and, do a few experiments, I think that would be fun.
And it's a little complex, right? Because if I'm looking at your data correctly, these cells don't come in right away. They come in later, actually after the mice have almost cleared the virus, right?
Yeah. Mm-hmm.
So,
So that's why we think that they're not going in there to clear up to do anything phagocytic maybe, but they're going in there and doing something else. We'd like to think.
Well, it's a typical post-viral inflammatory period, right? Like we see with COVID, you see a viral phase, and then it's in some patients an inflammatory phase, and maybe that's what's going on here, and it differs depending on the viral strain, as you saw.
Right.
Yeah.
But it is beneficial. It's somehow beneficial.
Yeah,
right?
It's somehow beneficial, and that's what we.
That's what you show in your next experiment, right?
Right.
Because there's more of these unique macrophages that are coming in from the serosal cavity in the milder ones, right?
Right.
So if the mice do worse, they're not recruiting in these cells.
Yeah, they're not recruiting them, or maybe they're recruiting them and the cells are dying.
And
so we haven't tested that yet.
So I think that this is actually kind of an important distinction that I was thinking about with the paper also, is that the idea is that mild infection recruits in these cells. It's not that these cells make the infection more mild.
Well, maybe they prevent the inflammatory.
Can you say
and that difference was sort of something I was sort of struggling with trying to decide.
Can
which
you say
direction.
Can you differentiate?
Yeah. I mean, you in the sense that, I think you can, because when we get rid of those macrophages, the disease gets worse. So I think that would suggest that they're doing something to help improve disease outcome. And so we actually titled the paper that way because it is we still don't have a mechanism, and I don't want to say that this thing comes in and is doing all this epic stuff, which is not necessarily true. But we do know that when they're not there, you get more disease. The animals lose more weight when they're infected with influenza virus.
Tell us about the depletion study.
So the depletion study, we use this molecule, this compound called clodronate, and it causes apoptosis in cells. And so if you put clodronate in a liposome, so we have these clodronate liposomes, the liposomes will get eaten up by any phagocytic cells, so including macrophages. And then once the dye gets into these cells, it forms these, what are they called? These aggregates that are just very stable within the macrophages. And it gives a very long-lasting signal, red signal. And wait a second. Did I just switch to talking about my dye?
Yeah, you just mixed up a dye, labeling them versus that's a PKH dye. Oh my God, yes. I'm sorry.
I went.
No, you were right. You were right. The clodronate liposomes, when they take them up, the macrophages die, and then they're not there anymore. Yeah.
Right. So they take them up, and then the macrophages get wiped out. And we made sure to do the experiment showing that they weren't wiping out macrophages anywhere else, because that was a big criticism from reviewers.
Reviewer 3. So, did you inject the compound into the pleural space?
Yes. Mm-hmm.
Okay.
Yeah.
Yeah, makes sense.
And then one day later, we look and we see that most of the macrophages are gone.
And then, which strain do you use of influenza virus for those experiments?
We use that pandemic H1N1 California.
Okay, the milder one.
The milder one.
And then when you deplete the peritoneal max, then you get more severe disease. Right?
When we deplete the pleural max, we get more severe disease. Yes.
Okay. I mean, could it be that the PMs, even though they're coming in after the viral phase, they're suppressing inflammation in a certain way. And that, if the severe disease is really an inflammatory disease, then maybe
Right.
they could be involved in that, right?
Right. That's what we're thinking, and that's why we really need to figure out our LPS and poly-IC experiments, because I think that would really show that this isn't specific at all to influenza virus, and if it has broader implications, that would be really cool too.
Yeah.
Yeah.
Do you know if there are these cells in other lung inflammatory diseases? So like COPD, or has anyone looked?
No, I haven't seen anything on that, but that's a very good question.
Because that's a chronic inflammatory
Mm.
thing, and if they're constantly trying to come in
Right.
to repair this inflammation, it might be an interesting model to kind of try and find them.
Agreed. I am going to look into that. I'm writing this down. Where is my pen?
Yeah, and thinking
Pen.
about
You still use a pen, Juliet? I thought you typed everything.
No, I actually really like taking notes by hand.
I do
I
too.
think it just, my brain works better that way.
Well,
Wait,
I think there
Julie-
are studies that show that.
Yes.
There are.
Juliet, maybe you need a microbe TV notepad.
Oh.
Yeah, that would make my day.
Sure. I'll send you some.
All right, please do. Yes.
One
so-
other.
Yeah.
Go ahead.
Oh, no. Okay. Yeah. So, yeah, we did that depletion, and we also did a depletion experiment using antibodies against the CD115, but that wasn't the best at depleting the macrophages.
Hmm.
Yeah.
Interesting. And thinking about migration of these cells, what do we know about their ability or inability to migrate there? Chemotaxis, what are they driving towards? Do you know any of that?
No, we do not know. So that's why I'd really like to. I'd like to do some Luminex and see what the the cytokines are
Yeah.
in the lung, and see if I can see any differences between conditions that could give us a hint as to what exactly is driving them there. Yeah, at this point, we don't know.
Right. And then for individuals who, would a deficiency be because they lack the differentiation factors to, do they have just fewer of them? That's one question. If they can get them there, are they less effective? Is it age dependent? Thinking of ways that if you have somebody who cannot clear an infection, or at least the infection is gone, there's no more viral replication, trying to make a small molecule of some sort to get those macrophages to the pleural space to help with repair. I mean, is that how you envision a therapeutic?
That is how I
strategy?
envision. Yeah, that is exactly how I envision it. Can we Can we get something that can specifically ramp up this pleural macrophage migration or whatever it is that they're doing once they get there?
Right.
Yeah, that is how I would think of it. But also, because the pleural cavity is such a small space and it's highly related to the peritoneal cavity, I was thinking also that maybe we could use the peritoneal cavity as a source of these macrophages to see if they could have the same effect if injected into the pleural cavity or into the lung. Yeah, so those are some experiments we'd like to do. We just haven't done them yet. Right now, I only have one student, so
yeah,
it's going to take a while.
Yeah, well, and it is interesting, like you said, being a virologist and now having this really cool viral immunology story. And then, I mean, that's what we do as scientists, right? We adapt to the question. So it's neat to see how you're migrating towards immunology. Yeah.
Yeah, so it's been a fun journey. I really love learning. So that was.
I mean, getting a PhD trains you how to learn new things, and you can learn anything after that, right?
Basically,
it shouldn't be no problem. So these are mice. Do you think the same thing happens in people, and can you ever find out?
I don't know if the same thing happens in people. I would think so. We have the same sorts of small macrophages and large macrophages, and we have lungs and a pleural space. But I so that's a lot of similarity right there. But no, I do not know if this happens in people. And what I really should do is try to find somebody who a physician or someone who works with mesothelioma or some sort of thing where they're really looking at the pleural space. That's the kind of expertise I think I would need.
So, Juliet, in mice, when the macrophages migrate into the lung, where do they go? Do they actually
that was
go
my
into the
question.
luminal space?
No, they
alveoli?
don't. They end up in the parenchyma.
Okay, because
space.
if they did go in the lumen, you could do a BAL, right?
Mm-hmm. No, they're not in that. They're actually in the tissue itself, and they look like interstitial macrophages. Is the phenotype they take on. Cool. So, people have been studying alveolar macrophages a lot, but there's a lot less known about interstitial macrophages. So that's also another area where I think there could be some space for me as well.
Yeah. But you could find someone who has patients who die of severe influenza and who are in a study, and you could get bits of lung and look to see if these cells are in the interstitial space, right?
I could, except that they have markers that are once they get into the lung, they take on this phenotype that looks like a lot like the interstitial macrophages. So we'd have to find out. We'd have to label them in some way, which you can't really do with humans.
No, well, you can't know who's going to die of flu either, right? So,
but can you identify them with unique transcriptome signatures?
That
so
was
even
exactly
if
what I
they
was gonna
have the
say.
same marker, then you could try and see by single cell or some other method to see whether they're there.
I have a grant on that. I've written a grant on that, and I just wish that they would give me some money to do this because
it's a great idea. So I think
exactly.
I was getting ready to ask the exact same question. So,
well, Vincent took my question because I wanted to know where the cells went. So I took your question.
Awesome.
Well, they're all pool of questions, right?
Yeah.
Well, funding is tough, as you know. Just have to keep trying. Don't give up.
Yeah. It's a persistence.
Yes. Yep. That's what I keep being told. So I
You're like, I'm hoping this
I will persist.
I will survive. There was a song called that, right?
Yes.
What are you doing next, Juliet, if you can tell us anything?
So aside from the pleural macrophage story, where I'm still trying to figure out what the trigger is for this movement, and I'm also trying to see if I can do things with the pleural macrophages ex vivo, like if we took them out and added different things that could send them towards an M2 or an M1 sort of phenotype, and then put them back in the animals, if that could make a difference. So I just want to do a basic experiment like that to see if this idea of modifying macrophages ex vivo could be useful at some point for treatment. I'm not sure about that, but I think it's interesting to look. I'm also go ahead, Brianne.
Sorry. So I've done a little bit with peritoneal macrophages, and you mentioned that. My question that I thought of is, how many pleural macrophages can you get from a mouse?
Probably not many.
How many? We about 500,000. Yes.
That's not terrible.
Yeah.
Cool. I just know about the challenges of getting peritoneal, and I wondered.
Yes. Yeah, it's not. No, we don't get a lot, but we get enough to do things with.
Okay.
Yes.
Cool.
Yeah. And what else am I interested in? I'd really like to get my dengue project back up. I'm also interested in STAT2 as a molecule outside of innate immunity. So there are some papers that have shown that people with mutations in STAT2 have these abnormally long mitochondria, and I have all I have all of these STAT2 constructs so I'd love to see what's going on there. And there are a few other things that I'm interested in doing, but right now I think that mitochondria thing and then getting back into the dengue. Oh, wait, actually I have a SARS-CoV-2 project as well, where we found that one of the proteins in SARS-CoV-2 is interacting with a couple players in the NF-kappa B pathway. And since, everybody knows that COVID has this like inflammatory component to it, I'm also interested in seeing if that this interaction could explain some of that inflammation.
Do you think there could be a role for pleural macrophages with SARS-CoV-2?
We want to do that experiment, so but we haven't yet. But I think so, because I don't think it's specifically the replication that's causing it. I think it's the damage and the response and the inflammation that's driving this movement.
That made me think about, you mentioned mesothelioma.
Mhm.
Have you thought about whether these are playing any role in cancer? Because that's another model where you have sort of suppression of immune response, and these seem to be dampening immune responses.
Right. So in mesotheliomas, they have found that the macrophages in the pleural space they take on that sort of they're really M2 and very anti-inflammatory, and so they're so they are also involved in cancer.
Cool.
Juliet, do you still work in the lab?
Very rarely. I never have any time. Whenever I start trying to set up experiments and something else pops up, and then I have to close everything down. But I am hopeful that maybe this summer I can do some stuff in lab. I miss it. Yeah, it's great to write this stuff up and it's great to analyze the data, but sometimes I miss actually just sitting there and doing a million plaque assays all day long. it just clears your mind. I would spend hours in the tissue culture room in Vincent's lab, just popping out, doing plaque assay after plaque assay. So nowadays, when my students are like, "Oh, I have so many plaque assays to do," I am like, "Oh well, when I was in Vincent's lab,
back in my day."
Yes, exactly.
I had a question about dengue. I find it the journey to a dengue vaccine very almost overwhelming. I just So from your perspective as somebody who's been thinking about dengue for a while, what do you think our public health strategy is going to be for that? Do you see a vaccine in the future? What are your thoughts, if you have any?
I'm a bit skeptical of us having a vaccine. I'd like to be hopeful about it, but it just seems like every time they come up with one of these tetravalent vaccines that the immunity is off for one of the viruses or two of the viruses, and I mean, for me, I think that's just really troubling for the most part. But if there is a way to balance that immunity, maybe I could see that working.
And by balance, you mean having the correct serotypes.
Exactly, having the same amount of antibodies to all four of the serotypes instead of, because so far what I've seen is that it tends to, you'll get antibodies for one and less antibodies for another serotype, and then that leads you to have immunity against one serotype, but not the other serotype, and we know that increases the risk of severe dengue.
And then I guess that goes back to, we'd have to know everyone's serological profile before vaccinating, and we just can't. That's just impossible to do. So, I know it's just it kills a lot of people, a lot of babies, a lot of kids. Yeah, and it's just it sucks that. Yeah,
it's really awful. So
okay, I didn't mean to end on a downer or anything, but I like to ask dengue people their thoughts, because it's a hard one.
It is.
Yeah.
All right. Anything else, folks, before we say goodbye? Everybody happy?
Yeah, this is great.
Yeah, this was lovely.
This was really cool.
Thank you so much for inviting me.
Absolutely.
Yeah, this was a really fun conversation.
All right, that's sorry, go ahead, Brianna.
I was just going to say, I'm now imagining experiments with some of the dyes that you use. So.
All right, that's immune number 76. Show notes at microbe.tv/immune. You can send us questions or comments, immune@microbe.tv. If you enjoy our work, please contribute, microbe.tv/contribute. Our guest today, Juliet Morrison from the University of California, Riverside at JUMO DR. Thank you, Juliet.
Thank you so much. Thank you so much, Vincent, Brianne, Cindy, and Steph, for a lovely conversation.
You ever come back to New York, Juliet?
I want to. One day. I miss the East Coast. Yeah.
Well, you have
Southern
better
California
weather.
is yes. The weather is nice here, but the East Coast is closer to Jamaica.
Right.
Yeah.
I remember your mom was a nurse, right?
No, hairdresser.
Okay. Why did I think nurse? And I thought that was your source of science info, but no.
Mm-mm.
Okay. So does she have anything to do with your hair now?
No, it would be so much nicer if she was.
I don't know. It looks great to me.
Well, thank you.
You're welcome. Cindy Leifer's at Cornell University. Cindy Leifer, I don't know. You guys are on X or Twitter or Blue Sky? Are you on?
Yeah, well, I'm going to join Blue Sky. I have some
But you're
cards,
on
but I
you're on
haven't done
X
it
right
yet.
now,
But
right?
Yeah.
And so are you,
still, yeah.
Juliet, right?
I am, but I'm considering Blue Sky as well. I'm just waiting for more people to be on it.
Okay. Thank you, Cindy.
Yeah, thank you.
Steph Langle is at Case Western Reserve University. Stephanie Langle on X. Thank you, Steph.
Yes, thank you. Thanks, Juliet, for coming.
Thank you.
And Brianne Barker's at Drew University. BioProf Barker on Blue Sky, right,
Yes.
Brianne?
I still have the X. I just don't really look at it.
Hmm.
The Blue Sky I'm getting into.
You like it?
Yeah.
Cool.
Yeah, it in a lot of ways it reminds me of kind of what science Twitter was like before.
Ooh,
Okay.
okay.
So I've been enjoying it.
I'm motivated.
Me
I'll do
too.
it.
Yeah, I like it. So I would definitely recommend it. It's just tricky sometimes to get invites, but there are ways you can get invites if we need to get them. And thank you so much. And Julia, I learned a lot.
Yeah, thank you.
I'm Vincent Racaniello. You could find me at microbe.tv. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.